A fully mature sperm cell is uniquely designed for its job. Its tail, which is called the flagellum, that's singular, flagella is plural, it's the only true tail on any cells in the body. Rather than moving in a whipping motion like we see in textbook cartoons back and forth, this tail, it actually spins like a little propeller. Around the base of the tail, there are tons of this mitochondria here that are arranged in a spiral pattern, and these produce ATP to facilitate the movement of the flagellum. The energy is essential for the very long swim that sperm must take through the female reproductive tract. At the front of the sperm in the head, we find this pouch called the acrosome, and this is a specialized structure, kind of like an enzyme-filled cap, that helps the sperm break through the outer layers of the ovum during fertilization. The rest of the head contains some tightly packed DNA that's going to combine with the DNA in the oocyte, and this nucleus here, it only contains 23 chromosomes because it's going to combine with 23 chromosomes from an oocyte, right? And that's going to give the resulting zygote the 46 chromosomes that are average for a human. Spermatogenesis is the process that takes a sperm cell and makes about 300,000 sperm per minute, or 400 million sperm per day. It's a lot of sperm. When we talk about making sperm, there are two different terms, and it's important not to mix them up. Spermatogenesis, with a T, is the process of creating spermatozoa. That's the complete process of forming sperm cells through both mitosis and meiosis. Then we have the process of spermiogenesis with no T in there. And that's the final stage when these spermatozoa become fully mature and capable of fertilization. Spermatogenesis begins with a round of mitosis, followed by 2 rounds of meiosis. As these cell divisions happen, sperm cells migrate from the outside or the outer edge of a seminiferous tubule in toward the lumen. So they move as they go through these cell divisions. While this process unfolds, the developing spermatocytes are surrounded and protected by nurse cells, also called Sertoli or sustentacular cells. That's all the bluish purple that you see in between the cells of this seminiferous tubule. The stem cells that are on the outside, they do mitosis, and one daughter cell stays behind to continue to be the stem cell, and then the other daughter cell goes forward and does meiosis, gradually turning into a sperm or a gamete. Once meiosis is complete, we have these 4 fat little cells down here that are called spermatids. These spermatids, they are not yet mature sperm. They typically don't have fully formed flagella, and they don't have the right structure for fertilization. So they need to mature further and go through that process of spermiogenesis before we can call them truly functional. A fully developed sperm cell is called a spermatozoon, and the plural form is spermatozoa. And this is also why the word sperm is not a great word to use. That could apply to these stem cells, these dividing cells, the spermatids, a fully formed one. It could apply to a sperm cell in any of these forms. Knowing that spermatogenesis is a series of cell divisions is the first step to understanding spermatogenesis. Remember that mitosis makes clones, right? Mitosis doesn't make a parent and a daughter cell. Mitosis gets rid of the original cell and makes 2 clones, right? And then meiosis takes one of those clones and it cuts the chromosome number in half, making cells called gametes or sperm. Cells called spermatogonia are positioned in the basal compartment of the seminiferous tubule. And these spermatogonia, they divide every 16 days or so using mitosis, right? So they replace themselves, and then the other clone that they have will go on to do meiosis. And actually, will go on to do meiosis. This clone, at first, is called a primary spermatocyte. The primary spermatocyte starts to go through meiosis. And remember, meiosis has 2 rounds. So in the first round of meiosis, 2 cells are created, and those 2 cells are called secondary spermatocytes. Could I make this even more complicated? But those are called secondary spermatocytes. They have half the number of the chromosomes as the original primary spermatocyte. And so they are no longer this person's cells. The body won't really recognize these cells. The secondary spermatocytes, they divide in a second round of meiosis, aptly named meiosis 2, and each of them dividing into these 4 cells that are called spermatids. There are 4 spermatids that remain embedded in the luminal compartment of the nurse cell. These spermatids will eventually break free and start to move through the seminiferous tubules to the straight tubules that lead to the rete testis, and then through those efferent ductules that lead to the epididymis. While waiting in the epididymis, spermiogenesis occurs. This is not a cell division, but more of like a maturity. It's more like the sperm cell is maturing. At first, a very small flagella develops, and it's anchored to the centrioles that are used in mitosis. The organelles involved in protein making, they get all broken down except for the endoplasmic reticulum. The ER becomes this sac of enzymes that positions itself opposite to the flagellum. And its purpose is releasing all of those enzymes to penetrate an ovum if it's encountered. There are mitochondria that start to arrange themselves in the mid-piece of the spermatozoon, and they take on that spiral sort of arrangement. And then the final spermatozoon has a head, a mid-piece, and a tail. And this vessel is the perfect vessel for ovum seeking.